Okay, everybody. My name's Larissa Froschel, and our lab was the Christner lab. We worked specifically with Christina Davis from the Christner lab. And what Christner lab does is they actually study microbes in subglacial lakes in Antarctica. So it's pretty cool. So their main project that's going on right now, they love to give them cool names. So their main project is called SALSA. And that stands for Subglacial Antarctic Lakes Scientific Access. And so we really kind of wanted to focus on the microbial life versus what kind of biodiversity of microbes might we see if we were just to test water around here versus what they might find underneath the lakes. And so we also, as many of the other groups did, we had people from very different age groups and grade levels and everything else. So we tried to make our lesson plan like this shell that then you could add in specifically. So we can, I guess, move to the next slide. Okay, so our learning objectives are pretty varied because we have anything from 4th or 5th grade all the way up to 9th and 10th grade. And we kind of tried to color code or at least box them separately so that you would kind of see. And I'm gonna kind of focus a little bit more on the 9th and 10th grade just because that's my grade level. So, okay, we can move on. As far as our learning standards, we wanted to focus on nature of science, but it's so hard to separate that from all the other standards, like all those content-based standards. And so I think our biggest struggle was deciding which standards to use because when we started deciding, you know, we want to look at life underneath a microscope and we want to look at, you know, all the different things that are there, compare and contrast, and we're going, there's like a million standards right here. So what we focused on was the nature of science standards first for making predictions and the hypotheses and then comparing and contrasting. And then we said, whatever other standards you kind of want to go for, you could make this lesson anything in that respect. So up at the top in the white, that's the 5th grade standards. And then in green are the 8th grade. And then the 9th and 10th grade, those are the regular biology standards that we have down there. We can move on. So as far as prior knowledge, this could change a little bit based on which direction you want to take it in, but absolutely it's going to be important that climate zones and their environments and what can live there are going to differ. And so also not only that, but different like aquatic habitats, whether you're brackish water, marine, or freshwater, that could affect. So yeah, we wanted to have those standards, but then also the nature of science, which you put at the bottom, for scientists use tools and techniques to understand living materials. So in that aspect, we really were looking at what were the techniques used in SALSA and how does that kind of bring science to life for the students in the classroom, meaning, oh, okay, so I know this SEM microscope, this TEM microscope, well, what does it actually do? How can you actually use it? So we really want to talk about the technology there as well. We can change the slide. Just some vocabulary here. It's pretty varied. Again, it depends on kind of which age level you're working with. So we can move on there. Our resources. SALSA has some great resources that you could go to, and we were planning on using some of those. We also, Christina showed us a really great online activity called Life in a Drop of Water, and it's a great kind of virtual. And so what we really did was we kind of took that protocol for Life in a Drop of Water, and that was our core of the lesson. And so then each of us kind of took it in a different direction from there, based on our grade level, but that was our basis right there. Dr. Christner also had a really great podcast that was put on by UF. And so I know that in some of the classes, we would definitely wanna have that podcast in there as well for information. We can move on. As far as resources, we would absolutely want to show our students some of these plates that were cultured by Christina Davis at the Christner Lab and kind of show them all the really cool colors. And we wanted to talk to them about different staining techniques as well. because see at the bottom, that's the cyber gold, which is something we don't usually use in staining. So I'm gonna talk about staining again in a sec. We can move on, but those, you know, we gotta show them those pictures. Okay, so materials, again, gonna vary, but if you kind of break it down, we all have that life in a drop of water, and then whatever our extension activity is, we kind of go out from there. We can move on from there. So 5th grade wasn't really my area too much, but what they want to do is focus on that life in a drop of water with the salsa video coming first. And then of course follow up with a scientist visit. And they were kind of looking at freshwater versus salt water and also different environments. So if you put the water in the refrigerator, the freezer, you have it at different temperatures, how is that going to affect what's living in that water? So that was the 5th grade. We can move on to the 8th grade. The 8th grade, they're taking a really interesting view of it. They decided after the salsa video to move on to an experiment for respiration for soil microbes, which is definitely apropos for the salsa project and everything that they're doing. So 8th grade took it that direction, and then we can talk about the 9th and 10th quickly. We can change. I'm very long-winded, as you can see. So for 9th and 10th grade, what we really wanted to do was have them work in lab groups, and we wanted them to start with their comparing and contrasting skills, so to speak. So they would swab their cheeks for some epithelial cells, stain them with methyl blue, and then onion cells as well, stain those with the Lugol's, and then they'll be able to just start comparing and contrasting right away. In addition, I love when the students look at prokaryotes, like prepared slides under the microscope. They really get a kick out of that and the different cell types and shapes and everything. So we wanted to put that in there as well. So those of you who are bio, you know we're hitting like a million standards right now. So prokaryotic versus eukaryotic, plant and animal structures, I mean, there's a lot of them. But basically, we just want them to be thinking critically. Our focus for them is making predictions, collecting data, and comparing, contrasting, and then interpreting that data. So I think the takeaway for us, which was really great, was it's really not hard to integrate nature or science into every aspect of what we do. I used to think like, well, how am I going to put that in there, you know? But now I'm kind of like, how am I not going to put that in there? Because I wanted to use every standard. So it was really cool to see that. After that initial microscope, that's day 1. Day 2 would be that life in a drop of water lab, which is actually a virtual, like interactive, and then we would also have them either collect water or we could bring water in for them, and they can look at what's under the microscope now, and now you're really critically thinking and comparing to, well, what's swimming around in there versus epithelial cells or versus the onion cells or the prokaryotes. And then we wanted to bring the virtual science, either virtual or in-person scientist in, and then we wanted them to kind of predict, the students to predict, do you think they found anything alive underneath the ice, you know, the glacier sheets of ice in Antarctica? And so we wanted them to kind of think critically there and then have that whole presentation. And then we also really thought it was important for the scientists to talk about their technology and their techniques of what they use, and they're, you know, kind of comparing, well, in the classroom, we're using a light microscope. Do the scientists use light microscopes too? And what else do they use and for what purpose? So again, hitting even more standards. But that was kind of what we were thinking for 9th and 10th grade. We can change the slide. And after this, basically we were thinking pre and post, some sort of a Google form, especially now with everything being virtual and stuff like that. So we really wanted to gauge the background knowledge, which is going to be a different level depending on where you take this lesson and what age group you're bringing it to. And the post activity, we kind of like differed a little bit on what we were thinking. Some of us wanted it to be more formal, a formal assessment here, comparing, well, these are the questions you got in your pre, let's take a post test now and just see that growth directly. Some people wanted to be more reflective about the summative assessment and just, you know, what have you learned here, and then, you know, be a little bit more specific, and then bring in the scientific method in there and stuff as well. So we were a little varied on that, but, you know, you would kind of make it what you wanted it to be for your class. And do we have one more? I'm not sure. We can change the slide if there it is. Yeah, so that's our project.